I'm just not sure how I'm navigating here. No, I just want to get out of this photo. There we go. And next. Oh, okay. Well, what do you want to say about this one?
Not much to say. I just found it this evening or late afternoon in my lawn while I was walking over to a neighbor's.
And you didn't open it up?
I didn't open it. I didn't do anything. I just took a picture. I thought, oh, cute, but it definitely looks a little oldish. I also, kind of, I think maybe pushed on it. Maybe you can see on the top.
Yeah. See, I was wondering if it was a young dead man's foot.
No, it's definitely a puffball. I've found puffballs there before, so I think it's just an older one, and maybe it's the famous group of spotted puffballs, like the next picture.
Okay.
No, it's just funny that. See how similar they look?
It looks like a granite river stone.
Well, it is, yeah. But it's so funny how these two kind of look the same. So, but yeah, I didn't look further into the real puffball one.
Yeah.
Yeah. And then these guys here. Oh, that's a funny thing, because I found that I took down my above ground pool and I had a tarp underneath to protect the pool, and that's what I found under the tarp. So, and I think that's also a puffball because these two little sack-like things that you see on top there, they look like they burst at some point and released spores. And yeah, this mycelium net underneath. I thought it was interesting.
Well, that's a great photo.
Mm-hmm.
I mean, it's to see the mycelium like that. So
yeah, and also that it just grew and matured under the tarp. It was pushed down by 5,000 gallons of water. So. And it still wanted to come out. So.
Cool.
Mm-hmm.
And you have one more, and then we're going to stop because we need to switch over to the regular meeting. Okay. Brown spored. That's
yeah, and it's like more like a rusty brown almost, as you see on the cap on the lower left there.
Oh yeah, thank you for pointing that out right there.
Mm-hmm.
That's my mouse isn't working that well, but right there.
Yeah, I'm not actually, I didn't see a mushroom there. I don't know if this top one, the one right above it, was closer to it at some point, and maybe they moved apart. I don't know what dropped out those spores on there.
Well, one of the things that was published recently, and I mean, I've puzzled over this for years, but it turns out with just the right wind, you get a backlash, and it's from this mushroom.
That it's the same, actually, the same mushroom.
Someone finally published it with like a little wind tunnel experiment.
Oh, yeah. Nice.
And they showed that they can, even though we think of it as one dropping on the other, which is how we usually get that spore print when they're like that, a single mushroom, it can waft back up and deposit on top.
Mm-hmm. Nice.
So that's probably what happened. And did you have an idea for the species?
Well, just I didn't know it all, but just what my gut spat out was Hebeloma.
Okay.
Which we talked about, I think, in the business meeting, right, on Wednesday. Didn't it come up?
Yeah, the bone finder.
Exactly. And so I just looked at my quick guide, and it said for rusty brown spores with adnate or gills, that Hebeloma would be a candidate. So I didn't look further into it, since it was just a couple hours ago when I found them.
Yeah. Mm-hmm. Yeah. Okay. Well, nice find. I mean, I love finding stuff like that, and uh, maybe you'll find some bones there in the future.
Yeah, who knows what's buried underneath?
Yeah. Okay, well, I'm going to stop there because we're going
Right.
to go. So I'm going to stop sharing, and I'm wondering if we have Vanessa. Vanessa, are you
Yes.
here and would like to discuss the mushroom fair? And if John, if you're here too, John Thrope.
I'm here.
Yeah, so I think you should both take a minute. Vanessa is the field trip chair. She
Or you mean the
is-
mushroom fair chair?
Sorry, mushroom fair chair. I don't know what I'm saying. Mushroom fair chair. And she is oversees the mushroom fair. She works pretty closely with John, who's the special event coordinator, and does the volunteers. And so if you're interested in helping and working with Vanessa, I think you would possibly sign up, depending on what the chore is. You might also want to sign up with John just so he can keep track of what you're doing. But we have some things in terms of the kitchen and other things that I think Vanessa and John would like to talk about that we need volunteers for the February 2022 Mushroom Fair, which will happen in person.
Yay. Yeah, so
Yes, it is yay.
yeah, I know. So exciting to be back. But we are planning our 38th annual mushroom fair. So that's going to happen Sunday, February 14th in 2022. So usually how we run the fair and how we run LAMS is everything is volunteer run. So for the fair, in the months before, we start making kind of like a fair committee. So if you want to be part of the fair committee, just email me. We're going to be needing a couple different roles. So depending on like your interest and your skills, we are going to need things like kitchen manager, kitchen volunteers, people to work the merchandise table. So people who are willing to help us sell LAMS t-shirts and things like that. So we'll need some volunteers for the merch table. We'll also need a contest coordinator and also a kids table coordinator. And then we're thinking of doing 2 new things this year. So we're thinking of doing a costume contest. So normally we always do an art contest and a photo contest, which are pretty popular. But it kind of came up recently that people do like to dress up like mushrooms. So we want to make that opportunity for people. So if you want to run the, costume contest, get in touch with me. I think that's it as far as like specific areas we need volunteers for. But if you do want to volunteer, get in touch with me or John. And then we're also doing a call for vendors. So if you own a mushroom related business and you want to sell your merch, just get in contact with me and you can find out how to vend at the fair. And I think that's it.
Yeah, I'd just like to say that, one of the hit features of the fair is that LAMS usually makes and sells mushroom soup. Usually a wild mushroom soup like wild porcini. And we need a chef. Past chefs have been Andre Kohler, who, if you were on for the mushroom slide share, shared with us. Kat Chang has been one in the past. And those mushroom soups have been to rave review. We've also had contributions like Candy Cap cookies and Candy Cap horchata, but the mushroom is the headliner, and making food and publicly selling it is a little bit of a challenge in terms of you need to have some knowledge of kitchen food practices for larger scale
How
cooking.
many portions?
Well, certainly 100 or more. Does anyone remember?
I think I usually like, how do you say, tried or went for 200 portions.
And it's what day again? February what?
13.
I'll
And
do
then
it.
there's actually also a prep day on February 12th, setup day. And I think Vanessa forgot to mention that. So there's also always volunteers needed for that also.
Yeah, the soup gets made the day before and it gets refrigerated and then heated up that morning. And with mushroom soups, I don't know if you know, but that process actually often enhances how they taste.
I mean, I'm a chef. I own a few restaurants, so I'll do it.
Oh, so we would love to have your participation.
Yeah, I'll be the kitchen manager. I'll make the soup. I'll block out my schedule that weekend.
That would be awesome.
Yeah, all right.
Very good. Thank you so much.
Sure.
I'm also happy to share recipes, but I don't think you need one.
Yeah, so just to interject then, we can work with you to get wild mushrooms. We've had various sources. You may have your own sources. You know, a lot of the time we have to wind up with wild dried porcini, but we're open to what you want to do.
200 portions, and is there a budget?
It can't be truffles.
Okay.
I think porcini comes within the realm. We can discuss it.
Okay. Yeah, I'll do it. It's actually easy for me. It's not.
Thank you, Jeremy. That's an awesome contribution.
I have
And you'll be really well appreciated too. So
I have a commercial kitchen that I can use. It's not that big a deal. So we'll figure it out.
Oh, well, so they have a kitchen at the Arboretum.
Okay, then I'll use that one.
And if you want to come, because it gives a chance for everyone to hang together and do stuff, but we'll accept however it works for you, certainly.
Okay, no, that's fine. If it's a commercial kitchen, Annabella, yes.
Yeah, it's a commercial level kitchen.
Okay. Yeah,
It's a certified kitchen.
Okay.
All right, and John, do you want to say anything? Do you want to maybe put your contact in the chat and just make sure people can get a hold of you? Same for you, Vanessa. I'm not looking at the chat, but if you could, just so they don't have to go too far. It is a bit of an operation because it has multiple facets and it's in person. We won't be commuting electronically.
So yeah, anybody who wants to volunteer for the fair can see my email in the chat. And if you have a preference for your position, just let me know in the email.
And John, let's follow up with your usual thing that you structured that shows like maybe some sign up times, because the fair is a, most of the day event that opens at 9 and closes at 4. So no one wants to volunteer to do one thing, for 7 hours in a row. If it's working one of the tables, usually that people do it for a couple hours, for example. So please email John, and you'll also see it come around as an email, as a LAMS announcement.
Yeah, just keep an eye out for the email and we'll set up a schedule so everyone can pick a time.
Okay. Well, that sounds good. Thank you for making those announcements, and you've already been successful. So I'm really happy that Jeremy can do that. Anything else that we need to discuss? Let me just say that we were hoping to announce a location for in-person in 2022, and that is not ready yet. So we thought we were close and it didn't quite happen. So we're still working on it. And I've got some help from Aaron now too, that I'm definitely hoping we get forward on pretty soon.
I guess I want to throw out quick that of all the members, if anybody is good at like personal assisting, I think David could use an assistant. I think he has a huge workload. And so if anybody is up for the task, please let David know.
Well, you know, one thing that really helps is Aaron already volunteered to do, to help track down the other locations, because I thought we were there. We have one backup that we haven't agreed on yet, and Aaron's helping with those other ones. And that was really my big bottleneck, was I just wound up with a crunch at work and not being able to pursue that as much as I wanted to. So we're trying. And thank you, Andre. I appreciate that. Any other announcements?
I'll just chime in. Our next, the next reading group meeting is actually next Monday, and we are finishing the discussions for the mushroom at the end of the world. And yeah, if you haven't, I'll send out another email tomorrow on that. If you haven't had a chance to read all of it, don't worry, come in and join and check it out. It's a really good book. There's actually a feature film in the Fungi Film Fest this week that featured Anna Singh. It was really cool with Peter McCoy, called Mushroom Speaks, which I also recommend checking out. But come and join us, Griffith Park, next Monday, and I'll send out more information about that tomorrow.
Thank you,
And in
Aaron.
the chat.
Yeah, that sounds like it's been really successful. So anyone else want to make an announcement? Well, if not, I'll go ahead and introduce tonight's speaker. So tonight we have Rachel Zoller, and she's from the Columbia River Gorge in Oregon. She's also known as Yellow Eleanor on YouTube. And she'll have to explain that to us maybe. I'm guessing it might have something to do with chanterelles, but that's just a guess. She's a mycologist and mushroom educator, and she shares her passion with people in her community. And tonight she's sharing what she wishes she'd known when she was a beginner mushroomer. So Rachel, thank you for coming.
Thank you. Thank you for having me. All right, so first things first, let's get the screen sharing working properly. And let's see if I remember how to do that. Here we go. How's that look on your end? Full screen, you see it?
Yep, very nice.
Excellent. So yes, my name is Rachel. I'll just get the yellow Eleanor thing out of the way first. How about that?
Sure.
So it is actually not mushroom related.
Okay.
Most people do guess that. It is a very nerdy reference to kind of Lord of the Rings, The Hobbit, Tolkien writings. It's a flower that the elves hold in like high reverence, and very into that kind of thing, Lord of the Rings. And so when I started falling in love with mushrooms, I started taking hundreds and hundreds of photos, and I wanted to start sharing them online, but I didn't want my actual name to be on there. And so that Yellow Eleanor sounds nice. So that's what I named my social media accounts when I started sharing my mushroom photos. And then it just became a whole thing after that. So a lot of people know me as Eleanor, and when they realize that's not my real name, it's kind of funny, but I just stuck with it because I liked it. And there you go, now you know. But yes, I am in the Columbia River Gorge, and I am on the Washington side. I live just a few miles down the street from Michael Bugh, who was your speaker last month. And I'll just say we're very opposite. We're friends, and I learn a lot from him. But he is so up there in academia and can speak science so fluently. And I see myself more as this girl who likes to play in the mud and look at weird things on the ground. And I don't come from an academic background. I didn't, my eyes weren't really open to mushrooms until after college for me. And so my experience was very much coming from just an average lay person trying to figure out this world of mushrooms and identify them. And that started for me about 10 years ago. And at that time, even 10 years ago, so much has changed. Now there's a lot more resources out there. Mushroom clubs, mycology societies are more popular. Foraging in general is more popular. But back then, it was like you take a deep dive where you don't do it at all. And I'm sure there are a lot of folks who kind of share that similar story as well. So to get started, I'll just go from there. Like I said, I didn't study mushrooms until after college. And for me, it was just an experience of walking in the woods with a couple of folks that were kind of more friends of friends. And there was one person in that group who knew just enough to identify chanterelles. And their explanation to me about the scent, the texture, the weight of it in your hand, all those things I just had never considered looking at a mushroom that way. The rest of that hike, it was kind of as if the forest floor came alive. I saw colors and textures that I had never experienced before. Honestly, it felt to me more magical than, you know, those spring hikes that you go on where there's the wildflowers just totally bursting out in bloom. It's like what it felt like to me, because I grew up in the Pacific Northwest, hiking, hunting, backpacking, being in the woods all the time, but I never paid attention to the mushrooms until that day. And so I took home some of those chanterelles and they were very tasty. And I was like, I need to do this again. I need to learn more. And a few weeks later, I went out and again, another experience of just the forest floor being alive. And I took this photo that's on the screen right now and just took it with my iPhone. That's what all my photos are taken on. But it was this experience of looking at these tiny mycena on this mossy log and just being astounded by their beauty. And I said, either out loud or in my head, I don't remember for sure, but it was a question of, what are you doing here? And for me, that was the switch. It wasn't just what can I go out and eat, but what can I learn? Because I realized I knew so little, not only about what they were called, but what they were doing, what their role was in the ecology and the environment. And so that's when everything snowballed for me, and I just dove in head first and muddled my way through the process. It was a couple of years before I really connected with other mushroom people. It was at least 2 years before I realized that mushroom clubs existed. And just listening to you guys in the pre-meeting and everyone who's sharing photos and everything, oh, I love it so much. It's like, that's one of the best ways to learn. And you guys do a really amazing job with those photos that you send in and the discussions and the things you're learning. So it's very impressive. I was happy to listen in on that. So that's a little bit about my journey and how I got started. So what I'm going to share today, for a lot of you, it's stuff that you've known, you know, for years and years and years. But I do hope maybe in the way that I share it, it will be at least entertaining for you. But I always know there are people out there who are just like me, who, when they picked up their first field guide, were really overwhelmed by, you know, just what they were seeing, the abundance in the forest and some of the language and the terminology. And so that's what I'll be talking about a little bit. To get started though, here. I always like to start with an acknowledgement, a land acknowledgement, just where I am located here. I'm on the traditional and contemporary lands of Klickitat and Yakima people. So I learned to forage mushrooms between Pato and Lewitt. So that is Mount Adams and Mount St. Helens, if you were to look on a map, and nestled right along the Nike Poon River, which is the White Salmon River. So if you've ever heard of White Salmon, Washington, that is where I'm at. And I just wanted to recognize and affirm, you know, the indigenous, their ancestral and their ongoing relationship with the land, because it's because of them that I get to benefit and have my relationship now with the land and with the mushrooms. And also just a thank you to the indigenous communities around the world who currently protect 80% of the Earth's biodiversity. I feel like that's really important for us to mention right at the beginning, because the biodiversity just with mushrooms alone is outstanding and so beautiful and so wonderful. And I feel very grateful that I kind of stumbled upon, you know, mushrooms and got to be so connected right away. So I'm gonna start super simple here. How you get started in mushroom foraging, in case this is one of your first mushroom events that you're attending or lectures that you're attending, I just want to encourage you that it's super simple to get started. All you need is a basket or a mesh bag. Try and avoid plastic. You need a knife and a brush for cleaning and a good field guide. I love that about mushroom hunting, that it didn't take much for me to like fully get obsessed, because unlike skiing where you have to like buy the gear and pay for lessons and spend a lot of time out doing it only to find out you really suck at it, it's not like that. It can be really, really simple. So you get to do this and not even spend a lot of money or time investing into just diving in. Picking a field guide can be a little bit tricky. So I just want to give a few helpful hints. Actually, where you guys are in LA, you have some great mycologists right around your area who are writing amazing books. So you guys are very lucky. But picking something locally focused is going to be really helpful. If you have a mushroom guide that's covering a broad span area, it's not going to have some of the odds and ends and the special things that grow in your area. It's also going to be harder to muddle through hundreds and hundreds of species that don't even grow in your area. So locally focused is great. The more recently published is also helpful. The mushrooms themselves don't change, but a lot of the scientific names have changed, which, you know, you publish a book and 3 months later, I think that's what Noah was complaining about when Mushrooms of the Redwood Coast came out. And that's just what happens. It's kind of the beauty of the science as well, because it is such a young science. Then you can upgrade. Upgrade once you sharpen your skills. Maybe there's a certain family of mushrooms that really piques your interest, or you're really interested in the tiny weird little things and you get an Ask a Mycete book or something like that. You can always upgrade, and the more books the better. Who are we kidding? You know, that's just how it goes. An edible mushroom book, like something that's specific for that, is great to couple with a field guide. A field guide is going to tell you, you know, the names of anything as you're going on a walk. If you want to know what's growing in your backyard, a field guide is going to be helpful. But then if you want to hone in your skills on some specific edibles, the ones that are the most tasty, and just learn more details about them, how to prepare them and how to preserve them and stuff, an edible focus book is always a nice thing to mix in there. And this is the one I really enjoy. I will say, just I want to say, when you go online, choose your online resources wisely. Mushroom Expert, iNaturalist, Mushroom Observer are always great go-tos no matter where you are in the world to learn information, identification, and have others help you. Like I said, field guides are great. Online can be scary because all of a sudden you're opening yourself up to the whole world. And just say I've seen way too many examples online, especially myself. I do a lot of educational stuff on YouTube and on Instagram, and folks will post photos and get people chiming in on what they think it is from all over the world. And area specific information is very important. And unfortunately, there are a lot of people who are convinced that they're right. And you know what, that's going to happen anywhere you go. But choosing an online resource that is really dedicated to, you know, high quality information is very important in the beginning. And like myself, I remember sitting in the woods finding a mushroom and hoping I have enough cell service to like do a Google search for, you know, white mushroom with gills, which who am I kidding? Like I didn't know anything at that point. That's just what I thought you needed to do. But I learned shortly after that there needs to be some good resources in my pocket. So if you have a basket, a knife, a brush, a field guide, that's what you need to go out. Now what do? You get to go out into the woods. You get to see what you can find. You don't have to go to the woods. You can go to a park. You can go to your backyard. You can go to your neighbor's yard if they have fun mushrooms growing. Just ask permission first. But I do want to encourage you to start simple. this picture here, I'm sure it looks a lot like when you guys used to get to meet in person, or even what your mushroom fair might look like. This is the Oregon Mycological Society. This is the fall event that we used to do. We'd go out to the Oregon coast for a few days and just collect a variety of anything and everything to identify and catalog things. And this is just a few of the tables. There were at least 10 other tables with specimens on them from our few days that we were there. And when I started out mushroom foraging, I thought that's just what you did, that you just filled your basket to the brim, brought it all home and sorted it there. And it was incredibly overwhelming, not to mention a mess in my kitchen. And I learned very quickly that when a field guide or an online resource describes a mushroom as putrid, I didn't know what that word meant until I learned about mushrooms. You leave them on the counter overnight and you get some fun surprises sometimes. So don't collect everything you see. You know, pick and choose a few that look interesting to you. And when it comes to edible mushrooms, also just choose wisely. Pick a few to learn at a time and learn them well. Every mushroom is going to have a few other things that look like it. That's why we're always very cautious when we're identifying our mushrooms if we're going to consume them. It's important to learn all that. So if you want to go out and learn 10 edible mushrooms your first year or so of mushroom foraging, you got to consider there could be, 3 to 5 mushrooms that potentially look like those. So all of a sudden there's 100 mushrooms that you need to learn. And there's a lot of margin for error there. So you got to be cautious with that. The last thing I mentioned here is understand what a fungi is and the anatomy of a mushroom. The anatomy of mushroom, for some of us, can seem really simple and basic because we've been doing this for so long. But something that I learned in teaching foraging and identification courses where we got into the woods, I had a lot of folks coming to me who hunted chanterelles and morels, of course, two probably most popular edible mushrooms. And they've always just maybe grown up foraging those two, or those are the only two that they cared to learn at first, and now they're interested in learning more. And I used to have this expectation that folks who hunted those two mushrooms would understand how to compare and contrast other mushrooms and learn more edibles easier. And it just wasn't the case. I realized a lot of folks are so hyper-focused just on those edible mushrooms that they never took the time to learn the anatomy of a mushroom and how to compare and contrast so much other variety that's out there. So if you can get really comfortable and familiar with the anatomy of a mushroom to start off with, it really just gets you up another level to have a deeper understanding and appreciation, and also a clearer picture when you look at your field guide when it's asking you questions or pointing out features. So just real briefly, I'm going to go over this. I loved, again, at the beginning of the meeting, you guys were talking about the different things you were finding, and you were taking the time to break apart some of those more technical terms that can seem really cumbersome for beginners. And I really loved and appreciated that. I wish I had that. I wish I had that at the beginning of my journey for sure. But when it comes to the cap of the mushroom, it's the cap. Most people know that. You see them in the grocery store, you usually know it's the cap of the mushroom. But there's so many details and features. And all I'm going to say about it is take note of the details. How does it feel? How does it look? Is it sticky, slimy, velvety? Does it have tissue on it or peeling off it? Are there color variations? And there are so many fun words to learn to describe those kinds of things. But in the beginning, it's okay if you don't know those words. Just take the time and the effort to make the observation right there in the moment. And maybe take some notes. If you can put it into your own language, it's easier to translate some of those more scientific terms and descriptions later on. Cap shapes can be important, but I also always encourage beginners that if it is challenging for you to remember these kinds of terms, it's okay to let it be at first. Sometimes I joke about this. You have conic, convex, and the rest is confusing. That's what I like to say sometimes. And it's just really also important to know that a mushroom can look one shape one day and totally different the next because of the morphology of it as it grows and as it changes. It's just important to know that as detailed as we want to be and as exact as we want to be, there's always the flexibility of what's happening in the environment. And that's what makes mushroom hunting and identification a challenge, but also a little more fun in the end. And when you have groups come together and you can all pitch in and try and figure out what you're looking at. Then we have what's going on underneath the cap of a mushroom. These are where the spores are produced and released from. So we also call this the spore-bearing surface. You can have gills. You can also have false gills. There's no hard line that defines what would make something a true gill versus a false gill, because there are some that kind of look like a mix between the two. But overall, a true gill is going to be thin, blade-like, and go deep. And false gill is going to be shallow, cross and fork and vein, look a little bit like, I don't know, like gills have been melted like wax against the rest of the mushroom. You're going to find things like spines or teeth on the underside of your mushroom. This is another spore-bearing surface. You're going to find things like pores or tubes on the underside of your mushroom cap. All of these features can come in a variety of colors and textures as well. So you're not just going to use your eyes to observe, you're going to use your fingers to feel, run your fingers across it, give it a squeeze, the texture, if it's slimy, as many details as you can notice. There we go. All right, then next let's talk about the stem, which again, another feature that most people can identify just because they know a cap and a stem, two easy ones, but the stem can have some of the most vital information about a mushroom on it. You can just tell by the photos, the variety of things that you're gonna see. You see the top left one has a little bit of a blue staining going on it. The bottom left is a very bulbous base. One's very hairy, one's very chubby, one's kind of wiry, all sorts of ways that you can describe. My favorite feature on some mushroom stems is reticulation. Reticulation is like a finely netted pattern that you'll see on the bottom of some mushrooms. You can see it there. Here's a really like predominant example of reticulation on that mushroom there. All sorts of fun words and ways you can describe them the more you get to know them. The next thing I want to talk about is what's going on with the ring or the skirt. So if you've ever heard those terms or have looked at a mushroom that looks like it has a skirt, what this is tissue that's left over from a partial veil. A partial veil covers the gills when a mushroom is young. Then when it breaks away, it leaves behind a ring on the stem. So you can see that photo on the left, that partial veil is fully intact. Mushroom opens up, matures, and leaves behind that ring. Not all mushrooms are going to have a ring or a skirt. It is going to be up to you to make the observation as to whether or not there was a partial veil by looking for the evidence. This is an important feature on the stem that you need to look for. Like I said, some of it is going to be obvious. That mushroom on the right, it's got a nice flowy mini skirt going on. The mushroom on the left is of the Cortinarius family, and the Cortinarius, sometimes they barely leave any evidence on the stem from their partial veil. Their special kind of veil is called a cortina, and you can tell by this photo that it's very cobweb-like, almost cottony. And as it's breaking away, it's really just leaving more of a tuft of tissue. Sometimes we call this a ring zone. So looking at that helps you see that you do need to take super careful, detailed looks at the stem of a mushroom. You can see on this tiny little one that there is just a little fluff of tissue that is the ring zone. Not much evidence that's there, but the more you look, the more you'll start seeing those evidence. And in case you don't get the reference, that mushroom is on the tip of my finger. The bottom is my finger, so that's how tiny that little thing is. All right, so then you're gonna move on to look for evidence other places on the mushroom where there was potentially a universal veil. So universal veil encloses the entire mushroom when it's young. And it kind of makes the mushroom look like it's growing like out of an egg at first, or like it's a weird egg growing out of the ground. This tissue, yes, covers the whole mushroom. And as the mushroom breaks away, it can leave behind tissue in a couple different places. This mushroom here is just a really classic example of leaving behind a sac or a cup at the base of the stem. Now, just like with the partial veil, not all mushrooms are going to have a universal veil. Some do and some don't, and you need to look very carefully on your mushroom to see if there's evidence of it. So here's a good example here, where the sac is not always obvious. That tissue, that universal veil tissue, could sometimes be really thick and rubbery, or it can be really soft and cottony. And that will depend on what mushroom it is. And as the mushroom matures, it'll leave different types of evidence. On that amanita that's on the right, you really just see fluffy tissue around the base of the stem. It's not a very predominant feature and easy to miss if you're not paying attention to the details, where one on the left, it's pretty obvious that it's growing out of a sac of tissue. Also, the universal veil may leave behind tissue on the top of the mushroom. Again, on the left, this mushroom had a very thick and kind of like leathery universal veil. So as the mushroom pushed out of that, tissue just kind of fell off the top. You know, it didn't leave any evidence. Whereas the mushroom on the right, that amanita there, the veil was a little more cottony or sticky. So as the mushroom pushed out of it, it left that tissue on the top. So both the top of the mushroom on the cap and also the base of the mushroom at the stem may have evidence of that universal veil. Here's a really great classic example, the Amanita muscaria. And it's just so much fun to bring people out into the woods and find this mushroom and explain, you know, like, this is not a red mushroom with white polka dots, like actually on it. It's just a white or a red mushroom that has white pieces of tissue from that universal veil. And this photo is a great example of that, because you can see that the mushroom on the right matured quicker than the one on the left. And the friction of it growing up against it wiped off some of those warts, some of that tissue. It's a really great example. And then if you're paying attention closely to the photo, you can see that this mushroom also has evidence of a partial veil. It has a little skirt, a little ring on the stem. When you find this combination of a mushroom having evidence of a universal veil and a partial veil, it's really good to stop and pay attention, because potentially you could have an amanita mushroom, which are some of the mushrooms in that family are quite deadly and can be mistaken for some choice edible mushrooms. So it's important to learn those features so you know to keep an eye out for them if you see them in conjunction with each other. So that always begs the question of, can I touch it if it's toxic? Which I do want to mention, the Amanita muscaria can be edible if you prepare it right. It can be toxic if you don't. It can have, you know, psychoactive things happen with it. It's a fun mushroom that kind of like breaks all the boundaries when you want to try and categorize things, which also brings up a lot of contention and I'm sure fun conversations in the mushroom club world. So sorry, throw that one out there for you guys, but you can talk about that later. But can you touch it? Yes, you can touch it. There are very, few cases where folks have reactions to touching toxic mushrooms, and usually those are more of a contact dermatitis where they're allergic to something in the mushroom. There is one mushroom that I know of, it's more of in a subtropical area, that can really harm you when you touch it. It is very rare, and so I think you guys, where you are in California, don't have to worry about it. I'm actually not quite sure its range, but by and large, you can touch your mushrooms, even the deadly ones. It's important to get to know mushrooms by their feel. And so I always encourage that. And this is one of my little dudes. It's his favorite mushroom to find. So that's kind of your basic anatomy of the mushroom, but looking down at the very bottom, we have mycelium. So this is what's going on underground. You've probably seen it before if you're like raking up leaves. It's usually on rotting leaves within logs, or even if you're digging in the soil in your garden, you may have seen this as well. So mycelium, it's not the root of the mushroom, though it does look root-like. It is the vegetative part of the fungus. So it is basically the fungus itself. It is the entire organism. And mushrooms, they're just the fruit of that mycelium. They're the fruit of the entire organism of that fungi. Not all fungi produce mushrooms. If you think about like toe fungus and bread mold, they don't produce mushrooms, but it would be a great study. If you ever have mushrooms growing on your toe fungus, let me know, because I study that. So by logic, all mushrooms are fungi, but not all fungi produce mushrooms. Mushrooms are the reproductive structures And they produce by releasing spores. I already mentioned that. So they produce this great variety again that, I mean, just, I feel like we are very lucky, those of us that live on the West Coast, to have the variety that we do. Just, it's truly incredible. I always like to mention this as well, just to show the difference between fungi and plants. Fungi can't make their own food, so they're considered heterotrophs. That is what we are as well. We can't like create food out of sunlight like plants can. So fungi don't photosynthesize. They have to hunt, forage, or mine for their food, and they do all of that. Fungi, their cell walls have chitin in them, and there are a couple of animals that do that as well. Some of the crustaceans and some beetles, and plants, they have cell walls and cellulose. Both things try and help it have structure, but very different. Fungi, like us, they take in oxygen and release carbon dioxide, where plants do the opposite. And it's really beautiful to think of like the similarities that we have with fungi. And then it starts to make sense how we are more closely genetically related to fungi than fungi are to plants. And it's just a really neat thing. And one thing about this that I always love to talk about is the contrast of what, I guess, what a young science mycology is, and how cool it is that someone like me and people like you who maybe don't have an academic background can still participate in community science, in submitting specimens and getting photos and bringing in data, or even just being curious and asking the right questions. So mycology really is truly a young science. It was only 52 years ago that fungi were considered non-flowering plants. So it was in 1969 that they officially got their own kingdom, when Robert Whitaker, he was very interested in figuring out how to divide organisms more by their function rather than just their form. You know, fungi or mushrooms, you know, they grow out of the ground like plants do, and that's kind of why they got categorized in that. And it's easy to think, okay, 1969, 52 years ago, maybe that was long ago enough that they just didn't like have the scientific, you know, methods to really tell that fungi were their own thing and so different. But that's not the case. That's the point I like to make, is that in 1969, that's also when the moon landing happened. So there was enough science to send a guy to the moon for rockets and fuel and everything that goes into that. And yet fungi, which are constantly around us everywhere, just kind of, you know, hanging out as non-flowering plants. But it is kind of a cool thing to think that, oh man, there's so much to learn still in this field. And it's really fun in a way, because yes, as hard as it can be sometimes when these scientific names are constantly changing, at the same time, it's really cool and really beautiful to see, okay, this unfolding of just, okay, 50 years ago, non-flowering plants. No wonder why they're still needing to collect so much data and to learn so many things. It keeps changing. Oh, I already talked about this photo, so I'll skip that. But yeah, this is the photo that changed it for me, and I just so appreciate it. And it kind of goes into another thing that I wish I would have understood more when I was first learning to harvest mushrooms, and that's just the idea of how to do things in an honorable way. I was never somebody who was going in and trashing the woods. My dad raised me really well. He caught me once, I think I was like 10 years old, etching my initials into a tree. And I got in so much trouble for it that forever, it's like ingrained in my mind of like, you know, the questions of like, why would I do that? Am I bringing harm to the tree or to the woods? And that has stuck with me, so thankfully. But, you know, when it comes to mushroom foraging, there's sometimes the collection just for the table, but there's also the collection to figure out what mushroom you might have, the collections to bring to classes, to show people, to bring to mushroom fairs. And sometimes it can feel a little discouraging when there's these conversations going on, or you're seeing folks who leave behind a lot of trash in the woods. And honestly, for me, Robin Wall Kimmerer, in her teachings, she talks about the honorable harvest in a couple of her books. And it's just been really good for me to have something to kind of fall back in and questions to ask myself. The first point is be accountable as the one doing the taking. So I have to be accountable to the woods, the mushrooms, and to those who are going to forage after me as well. Know the ways of the one you harvest so you may take care of them. To me, this speaks to the fact that me understanding more about how the mushroom and a fungi functions in the woods, what the mycelium is doing, the more I know that and learn about not just what I can eat, but what can I learn from the mushrooms and about the mushrooms, the better I'm going to be at taking care of things. I'm going to do the right thing because I'm going to have that awe and that wonder of this organism. Ask permission, listen, and respect the answer. To me, this is reflection. I remember somebody saying that to me when I first started foraging mushrooms about you need to ask permission first and see what the mushroom says back. And I thought it was silly. I will admit that. I didn't understand the idea of just sitting with it and reflecting on it and thinking, oh, if there's only one here, you know what, maybe just leave it. Let's see if that's going to be the right choice here today. It just takes a moment to sit and reflect on that. The idea of never taking the first or the last is, for me, just a nice practice. I don't always follow it, I will admit that, but I think it's a way that, especially on a day when I'm going to go out and I know I'm looking for edibles specifically, it's a good practice to have. Again, another way of respecting and making sure that I don't go into an area and just kind of ravage through it. Leaving some for others, harvest in a way that minimizes harm. Using everything I harvest, sharing, not wasting. Give thanks and give a gift of reciprocity. For those of you that are all planning this mushroom fair that you have coming up in February, the book club thing that I heard about, that's super cool. I feel like all of that is giving back. All of that is reciprocity. You're taking knowledge and you're spreading it. You're encouraging folks to learn and understand mushrooms in a deeper way. And that is all a giving back and helping others understand things so that it will be respected. So when it comes to the collection process, keeping those ideas in mind, but we do need to, for sure, when we're trying to identify something, make sure we're harvesting it in a way we get the most information possible. So we need to look at the habitat. What kind of plants are going on around the mushroom? What kind of trees? If you're not super great at your tree ID, just knowing if it's hardwood or conifer can be really helpful. So that's like leaves or needles. You know, if you can get that, that's helpful. What is the mushroom growing from? Is it growing from soil? Is it growing from buried wood, rotting wood? Sometimes the mushroom might be growing on another mushroom. That's what's going on in this photo. Little tiny baby mushrooms growing off this old decaying one. So habitat is really important. And then making sure you unearth the entire specimen. Specifically, we talked about the stem of the mushroom and how much information might be on the stem, even at the base of the stem, if you're looking for evidence of that universal veil. So unearthing the whole thing is going to help you find that evidence. It could be attached to buried wood, which would be important information. There could be color changes in the stem as it goes down underground as well. Length of the stem is also really important. This Phaeoclibia species on the left, it was the first time I'd ever seen one before. It was my first like cool mushroom camp that I ever went to. My friend Travis found these and he like walked in the room so proud. And I love it because I remember it so vividly because I know that in the beginning of my mushroom journey, I would kind of yank on the mushroom and assume that I got most of it out of the ground. I didn't understand that there were times that no, you just don't do that. You use your hands, you unbury it and get as much detail as you can. Had Travis just kind of yanked on this mushroom and it snapped off and he assumed he got most of it, he's going to have a little brown mushroom. And as you know, there's a lot of little brown mushrooms out there, which would make it harder to ID. But he walked in the room triumphantly with that. And there are people across the room who could identify that mushroom because of that feature. So unbearing the whole thing to look for details will be really important. All right, then you get to pick on your mushroom, which is my favorite thing. As much as I love mushrooms, I pick on my mushrooms. Smelling your mushrooms, super important. 90% of the times it's gonna smell like mushrooms and dirt. That's fine, smell mushrooms and dirt. But your nose is gonna get really used to what mushrooms and dirt smells like. Sometimes it's gonna be fresh mushrooms and dirt or moldy mushrooms and dirt. But then those other scents are gonna stand out to you, that root beer, that bubble gum, the black licorice, the cinnamon, all those things are gonna stand out to you as well. Pinching it, bruising it, slicing it, breaking it. I didn't know I was supposed to do these kinds of things when I first started mushroom foraging. Was definitely not something I'd ever considered before, like especially with identifying, wildflowers. It's not something that you talk about. So breaking your mushrooms, sometimes they're going to exude liquid, and giving your mushrooms time. Sometimes when you break a mushroom and it exudes liquid, it may come out cream and then 10 minutes later be deep royal purple. All sorts of interesting things that might happen as you pick on your mushrooms. There's some bruising happening there. This picture was taken 10 minutes apart from each other. So this bruising is just an oxidation that's happening. Many mushrooms oxidize a lot of fruit. I think pear is a great example. When you bite into a pear, they brown really quickly. Same thing going on, except mushrooms sometimes have different colors going on. This one took some time for it to have that bruising. This one here happened instantly as I ran my finger across that soft, spongy pore surface. And this one here, I remember when this young lady handed this mushroom to me, at first I thought she cut herself with her knife because it was so blood red. Just really beautiful things that mushrooms can do when we pick on them. All part of the details, all part of the puzzle pieces as we explore and examine them. All right, so then after that, making a spore print is going to be something that's really helpful. So spore print is just collecting the mushroom spores in mass. You can observe their color. Why is it important? It's, again, just another piece of the puzzle. Spore print isn't an end all be all. So if it has white spores, you can eat it. If it has brown spores, don't eat it. It doesn't work like that. It's just another feature to add to the bucket. But sometimes I like to compare it to like going into a library and knowing what section of the library the book you're looking for is in. Makes it a lot easier because a lot of times spore color is one way that a lot of field guides will divide up their mushrooms or categorize them, but it's definitely always a predominant feature when it comes to figuring out the ID of something for an online resource or for in a book. It's a feature that's mentioned, so really important. If you've never done it before, very simple. You put your mushroom with the spore-bearing surface down on a piece of paper. If you have paper that's half black, half white, that's great because it could have light spores or dark spores. You can do it on a piece of glass or tin foil. That's also helpful. Covering it in a glass bowl is also a helpful thing because it keeps the air currents from moving the spores around too much. Also helps the mushroom stay a little bit more moist and encourages it to release spores. Wait a few hours, then take a look, see what's going on underneath. Sometimes a mushroom might be too old or even too young, needs more time, but there should be a spore deposit left behind for you, which is always a fun treat to see what you find. This mushroom here, I remember finding it and I made the assumption that it would have brown spores. A lot of mushrooms do. But I did a little hop dance when I saw that it had these kind of beautiful salmon pink colored spores, because the section in my field guide at the time, Mushrooms of the Pacific Northwest, it's like 100 pages plus for mushrooms with brown spores, 4 pages of mushrooms that have pink spores. So all of a sudden it narrows everything down so quickly. All right, so a mushroom's color will never determine its spore color. So you can't just make an assumption of what the spore color will be, just like I said. But there are ways that you can try and be a detective to figure out spore color even out in the field. I'll talk more about that in a minute. All right, oh, I guess I'm missing a slide. Whoops, now how do I go back? Just a moment. There we go. Oh yes, there we go. So once you make a spore print and you go through that process, that's when you can apply that information to a field guide, which can be a lot, but that's an important thing to do, especially if you're going to be eating something that you've never identified. And just a note about photography for identification. One thing is, don't just depend on a photo for a confirmed ID, which means like, don't just flip through your book and say, oh, it looks the same. I bet that's what it is. You'll want to look at the description as well. But this also applies to taking just 1 or 2 photos, putting it online, not somewhere that's not a mushroom forum. Don't just post it online and ask for information and have somebody identify it. It's just not safe. Taking informative photos is going to be really important. So all the features that we just talked about, the whole anatomy of a mushroom, try and think through that and make sure you get photos of any potential feature that a mushroom might have. You're going to benefit greatly because folks like all of you guys in your meetings beforehand are going to be very willing and ready to help when you have really informative photos. You're going to have the details for them to look at. They're not going to have to ask as many clarifying questions, but also it just makes it more fun, a little bit easier. So you'll get extra help the better that your photos are as well. And sometimes all it takes is 1 more photo to get the information that you need, or a lot more information. Just this photo here from the top, it can be a variety of mushrooms. But as soon as you get underneath, you see that it has gills. You see that the gills are decurrent, meaning they're running down the stem. You see kind of the shape of the stem, the fact that the stem looks a little fuzzy. The stem is attaching itself to some wood. So all of a sudden, there's more details there. That is the same in the case as this mushroom here. It's growing from a tree. It could be really tempting just to take a shot of how cool that mushroom looks growing from that tree. Get a photo of just the underside. Again, we see it has gills. We see the gills attached directly to the stem. We see it has a skirt. And if you notice on top of the skirt, it has a kind of a fine little powder. That's the spores. So you have a natural in nature spore print right there already. You know the spore color of the mushroom just because you have 1 more photo. So this is why we want to start simple and not collect everything we see, because that process can be very overwhelming. And study only a few at a time and pick only a few specific edibles. But then that always begs the question of, is it edible? You want to ask yourself that. What does edible mean to you? Does edible mean it just won't kill you? I guess that's a good place to start. But it doesn't mean it also won't upset my stomach. Is the texture and flavor generally desirable? I feel like I want to check all those boxes before I'm like, yeah, that's an edible that's worth pursuing for me. Because there are a lot of things out there that are edible, just like this one. I mean, it's super cool and fun to find and to poke and play with, but it's not something that I'm going to go out and collect for the plate because it's a gelatinous blob. Again, something fun. And yes, it's not toxic and you could consider it something to eat, but it's not edible as far as a choice edible goes. So as you're learning, pick something that's popular, because the reason it's popular is because it sits well for your stomach and it tastes really good. It has a nice texture. That's where you want to start off with. And then if you want to try this later, let me know what you think. When you're collecting mushrooms just for the table, and you already know what the identification is, you don't have to go through that whole process. You don't have to unearth the whole thing anymore, because you don't have to worry about that dirt and everything in your basket. You can just leave it in the woods, but you can pluck the mushroom or you can cut the mushroom. Either way you do it is perfectly fine. I know that people are very passionate about cutting versus plucking. And there have been a couple of significant studies done. One done right in my backyard. The Categorizing project was done over 10 years long. Through the 10 years and through the variety of plots that they had with cutting versus plucking versus not harvesting anything, harvesting partially, in the end, they found that it didn't make a difference for sustainability and mushroom yield year after year. So that study, along with the Swiss study, had similar results. And I like the phrase picker's preference. There's some mushrooms that naturally to pluck them out feels good, and that's just kind of what the mushroom does. There's some mushrooms where cutting it is nice because you leave a lot of dirt in the ground. But either way is sustainable, and that's just the point I want to make. So you do it how you'd like to, and if somebody yells at you that you're doing it wrong, you can say, that weird mushroom lady told you can do however you want. Clean what you can in the field. That way, less mushroom or less dirt in your basket, less dirt in your kitchen. I like to separate the edibles from other mushrooms. A toxic mushroom isn't going to like lean up on your edible mushroom and like rub its toxins on it. That doesn't happen. But mushrooms are fragile, and there could be a little mix up going on in there when you get to the kitchen. So I usually keep my basket for the edible mushrooms and then have like a paper bag for my curiosity and things like that. It's important that when you're going to eat a wild mushroom for the first time, there are a couple of guidelines to think about. I like the guideline of gathering it a few times before eating it. If it's something that has a lot of lookalikes, that's a good practice to have. Try not to overindulge too much the first time. You never know, maybe your tummy doesn't like it. Keeping a safety mushroom in the fridge is nice. I've had a lot of conversations with poison control out of Portland. They're closely connected with the Oregon Mycological Society. And, it's really a bummer that they have to look through your vomit to figure out what you ate sometimes. If you have a safety mushroom in the fridge, you can save them a lot of heartache. Also, just consider that psychosomatic symptoms are a real thing. So if you are really nervous about eating it for the first time, maybe just don't. Maybe wait until you're with somebody who's been foraging longer than you and prepares it for you. Maybe you'll feel comfortable then. Or if you have friends and family that you want to serve a wild mushroom dish to, and there's someone who just does not feel comfortable with that, don't serve them that. Because they can make themselves sick just from the stress of worrying about it. And that's not a good experience for anybody. The question of toxic, poison, or preparation, always be thinking about that. There's a lot of foods that we have, like eggs and raw chicken and stuff, and we just know how to prepare them to make them edible. And there are different mushrooms that sometimes need different preparation. So think about that as you're learning new mushrooms of what is the proper and best way to prepare this. For me, if it's my first time trying a new mushroom, I keep it as simple as possible. I dry saute it with like a little butter. That way I get to know the texture and flavor of that mushroom the best. But then you get to the point where you find things and you're like, this mushroom is not shaped like a mushroom. What am I supposed to do at this point? Well, if you look at this mushroom, there's a few clues, but quite honestly, if you were to like Google weird white mushroom that looks like it's bleeding, you would probably figure out what it is. But also there are some other features. If you look at the bottom of this mushroom, you see it has a few little tiny spines. This mushroom has spines on the underside. So there's the top, here's the bottom, there we go. We see it has spines. We talked about spines as being a potential spore-bearing surface. So you know you can look in your field guide for a section that has different spine mushrooms, and then you can figure out what the identity is. That's what's great about something that's a locally focused field guide too, or even any field guide. They usually have a fairly logical way of dividing up different species to make it less overwhelming. And if as a beginner, you don't want to learn specific species at first, but you just want to learn categories, this is a great place to start because there are some things that are just beautiful and amazing like this. It looks like a coral and it's called a coral. That's its just very broad common name, but it's enough to get you to a place in a field guide that you can start figuring out more. Corals and clubs are usually all grouped together. You have fun things like jellies, jelly fungi, I love them. And then there's things like cup fungi. And these things, again, are all going to be grouped together based on how they look. Elven saddles, morels, and other morel look-alike things. You have some things like this. This is a pig's ear, and it has false gills on the underside. So you're going to find it in a section with mushrooms that might look similar to that. This mushroom here is my favorite. This is the bear's head, Hericium abietes, and it's just a big old ball of spines, but it's still going to be in the spine section of a field guide in an area where you find that. It's delicious, it's edible, it's easy to ID, it has medicinal values to it. Got to be a top favorite. All right, then you'll find stuff like this, and I remember this one for me. I don't know why I didn't connect for a while. I remember being very frustrated. These are polypores or conchs, you can call them. When you look at the bottom of a polypore, there's tiny, tiny, tiny pinhole pores, and they are hard and woody. They feel almost the exact same texture as the rest of the hard woody mushroom. However, this is a lesson that maybe some of you can relate to. When I first read descriptions of mushrooms and they were saying like a surface is smooth or a surface is, large, I didn't know what to compare it to. Smooth compared to what? Large compared to what? And when it came to pore surfaces, I remember a description that I would read is that boletes have a pore surface that's easy to remove. Polypores don't. I was like, well, clearly these hard woody ones I can't remove. But then I would find things like this, and I couldn't tell if it was easy enough to remove the pores to be considered easy or not. And that may sound really silly, but when you're a total novice and you have nothing to compare it to because you've never looked at a mushroom before that way, it's totally normal to sometimes just not connect the dots. Another example for me was with turkey tail. False turkey tail is perfectly smooth on the bottom. Turkey tails are not. Well, for goodness sake, the bottom of a turkey tail feels pretty darn smooth in general. But when you compare them side by side, yeah, there's a little bit of a difference. Again, these kinds of things like the pores are easy to remove or hard to remove, or it's large, or it's stickier than normal. All these things come with experience as you handle more mushrooms, as you smell more mushrooms, as you play with more mushrooms. So just a few examples of how, those wires can get a little bit crossed at first because you don't have the experience and, that tactile information necessarily built up in your brain as a log yet. So if you want to get serious, I would suggest bringing a few extra supplies like wax or paper bags to separate specimens. Trust me, if you fill up that entire bulga basket with mushrooms and think you can sort it all out later and remember what mushroom goes with what, probably not true. Don't do it. Separate them, keep them finely, organized in there. Index cards or note cards are really great. I like to write notes on them. And I even will do spore prints out in the field where I will put mushrooms on the note cards, slip them into a bag, and then have them, set in the basket in a way that they can release their spores while I'm walking around. Sharpie is great. I get a ton of rain where I am. That's why I have to use Sharpies all the time because no other pens will really work out in the rain very well. A hand lens is super helpful to look at those tiny details. The stuff I talked about is just scratching the surface. There are things like looking at the edge of the blade of a gill to see if there's a color change or if there's speckles on it or how it attaches to the stem. So many wonderful fine details that a hand lens is going to make a lot easier to see. This is my biggest disclaimer. Please know you're making my head hurt. I just think mushrooms are cute and pretty, and I want to walk around the woods like an elf to enjoy the forest. This is me the majority of the time. I will admit to you, sometimes that's all I want is I just want to go and soak it all in. I don't want to have to think about taxonomy. And I don't want to have to think about these details to identify something. For you guys, I guess that first point doesn't quite apply. Join a mycological society. You guys know the benefits of that. I can't tell you how much that has enriched my life. Some of my closest friends now are people that I have met through the mycological society. And I've grown to understand there's so much more to mushrooms than just what you can eat. There are folks who are interested in mushrooms as natural dyes, cultivating them, just experimenting, all sorts of things. It's wonderful. Photograph your mushrooms instead of collect. You might even notice more details that way. I know there have been many times where I've taken some really up close photos or just a lot of angles on a mushroom and then later on recognized, certain details. In fact, in your guys' pre-meeting, there was a gentleman who shared that he took a photo of a mushroom years ago and he didn't know what it was. But now, through experience, he knows what a mushroom is. And it's fun. I have photos like that I took 10 years ago, and it's like labeled as random blue mushroom or something like that. And now I look back in my archives and I'm like, oh, I see that all the time. Totally know what that mushroom is. It's a great way to kind of like, catalog your journey. And for me, honestly, photography has helped me notice so many more details this year. There's springtails. I didn't know springtails existed until I found mushrooms, because I was never looking in places that I would ever see them. This year, this is a Cortinarius, purple one, Cortinarius violaceus, and those are the rusty orange spores deposited on its gills. Mushroom people often also come across slime molds. I don't know, it's gardeners and mushroom people are the only people who pay attention to slime molds, but you get to see some really fantastic details on the forest floor when you just want to walk around and soak it in. This mushroom, there's a little bit of a party going on. This is springtails munching on a mycena, while there's also a secondary fungus that's parasitized the cap of the mushroom. Things like bird's nest. I absolutely love bird's nest. They're super tiny, but they're just like, they're brilliant. Their whole life cycle is really incredible. They start enclosed, they open up, the rain drops down into that little cup and releases those eggs. Those little capsules there have the spores inside. Oh, this is great. I don't know if anyone's seen this before, but it's just shows that example of the rain falling into a bird's nest and having those capsules ejected. That way the spores can be dispersed better. In this photo here, this is another bird's nest, but zoomed in a little closer, that is my thumbnail on the far left. But you can see the little cords that are attached to each of those individual capsules. And on the end, opposite end of those cords is a little sticky substance. So when a raindrop drops into that, it pops out that little capsule. The sticky part attaches to something, kind of like a game of tetherball. That little capsule can now rest, decay, and spread spores better than if it were resting inside that. All the beautiful details on the forest floor. I started taking photos of mushrooms on my fingertip just to show scale. Because when you think about it, the more you learn about mushrooms and fungi and the power that they have and the influence they have on our life and the forest and the ecology, and all sometimes we see the evidence is that tiny. It's such a tiny thing that has such a huge, amazing impact on us. I love that. That's probably the smallest one I've found to date. That's a little mycena, and that is a Douglas fir pine or Douglas fir needle that it is growing out of. So sometimes that's all you want to do is go and enjoy the woods and enjoy the beauty. That's perfectly fine too. You don't have to look at all those other nitty-gritty details, but both can be exciting. And every time you go through the process of trying to identify something, you can learn something from it, even if you don't get the full actual ID of something. So just try and pick apart, your process and what you're learning. And every time there's going to be something that stands out to you. And I don't know, if you're really super new to the mushroom thing, I just hope that maybe after this conversation, you'll start to see mushrooms everywhere and start to notice those details. Take time to pick one up out of your yard, give it a smell, see what it might smell like. Just to, notice more details and appreciate that beauty, because we need more people doing that. Like I said, mycology is a young science, and there are lots of ways where folks like me and you can get involved in collecting data. But that's for another talk, another time. And I'll leave it at that. So thank you, everybody.
Thank you so much for sharing that. Yeah, I really appreciate that you're carrying people back to their beginning and helping reorient them and fill in what might be some gaps. We'll take questions. If anyone has some, you can put them in the chat. I don't know if you can see the chat.
I am pulling
and I'm seeing a lot of complimentary messages right now.
Oh, thanks, guys. That's really nice.
And if you want to ask Rachel a question, you can type it into the chat, or you can just, I think they can unmute now, Rich.
Anyone should be able to unmute.
Yeah, so you can just ask a question directly. So I see one from Jeremy. Can you further clarify how human beings are closer to fungi than fungi are to plants?
Yeah, so it's just through the genetic studies. So if you were to look at the map, if you've seen those examples of where things are in the evolutionary chain, genetically humans are closer from that divergence than plants are to fungi. Does that make sense? I'm not, like I said, I don't come from academia, so that's the best I can probably explain it. But yeah, genetically closer related.
So I'm just trying to read, they're going fast here. Something about slime molds could be used to run a maze.
Oh yeah, there's some great videos if you look that up. Yeah.
Yeah, you can find it on YouTube.
Mm-hmm.
Yeah.
That was really awesome. Thank you so much.
Oh, you're welcome. I was really nervous. I think Kate is the one moderating the chat, and she's a good friend of mine. And I had to text her beforehand about how nervous I was. So thanks, guys.
Oh my gosh, it was amazing.
You didn't need to be.
Also, I had a question. It's a little irrelevant, but I was wondering, because I know some fungi are kind of like going underground and turning into truffles and like closing in because of the dry environments, like some agaricus and things like that.
Yeah.
And like russula. If global warming continues and like climates get worse for them to grow and it gets just like drier and drier, like do you know like how like what percent of fungi would be like kind of going underground and turning into the truffle like things?
You know, I have no idea on the percentage, but I know from what I've seen, not personally, but just reading some studies, it does seem to be guild mushrooms that are doing that more than, but it could be because there's more guild mushrooms in general, and that's why that's happening. But absolutely, like that's going to become something that's way more predominant for sure. But percentages, I'm not sure.
So like would the genuses that like are doing that, so would there, so are like eventually, like hypothetically, if like I know russulas, for example, the new genus of those is like Macawanides, could like those ones that are going underground, like would like hypothetically would it be able to, for like all russula to become the Macawanides, or is it just
you know I'm not sure on that. You say that way better than I could, so good job on that. I noticed you speak your mushroom scientific names really well earlier when we were doing the beginning chat. I'm not sure about that. I mean, I think that's the question of like, the more we can learn and study about all the differences, like talking about that evolutionary chain and where the common denominator has been for different species. But looking forward, I don't think there's a lot that we can do to predict that for sure. But I love that question. And russula would be a really great genus to keep an eye on, because I think that is something that we would see a divergence from quicker than others because we're already seeing it. But I don't really have any answers for you for that. But wonderful question.
okay? Thank you.
I've got one from YouTube. Ian asks about your photography setup.
Oh yeah.
And, if you use like any enhancements on maybe your phone or whatever.
So yeah, it's my iPhone, and I've tried a couple of different macro lenses, and that's it. Just like slip-on ones. Nothing nothing very fancy, and trying to hold really still. So that's it. Yeah.
Holding really still helps.
Yeah. It's not breathing.
Then we've also got Angel asked a question of what are your thoughts about mushroom growing kits?
Oh, well, so I don't do any cultivation myself. I had somebody ask me about that at work and thought it was such a weird idea that they were seeing it in their catalogs for Christmas gift ideas for people. I think it's great. If you have people who aren't mushroom people yet, buy them a mushroom growing kit. A lot of places, a lot of cities, even in my super small town, we have a local like mushroom farmer. So the best you can to get as local as possible, because they've been doing the work to figure out what grows in the area best. Of course, they have controlled conditions, but they know what's going to fruit, you know, if they give you a mushroom growing kit. Or start really simple with something like an oyster mushroom.
And look in the chat. I don't know if they can see the chat on
hey, look at that. Yeah.
on YouTube, but LAMS has its own growing group.
Awesome.
Called GLAMS, and you can, which stands for the Growers of Los Angeles Mycological Society.
I love that.
David Roman Daniels is the contact, and if you can see the chat, you can find that there, or you can go to Facebook and find it that way.
Awesome.
Well, I have a question.
Okay.
I always hate to ask this, but it's one I always do. So what are the mushroom poisonings you see up where you are?
Probably the most common I see it is with morels being undercooked. Yeah,
Okay.
it's a really popular mushroom, and I think that is fairly common, widespread. And what else? I don't know if this would, I see a lot of folks eating Russula without doing their due diligence on it. And even though most of those aren't going to do much to harm you, but that's the mushroom family that I will get photos of and ask what it is after people have already eaten it. So
Wow.
I think it's because it's somewhat robust. It looks nice, but that is not a good practice. We'll just say that. Yeah,
okay, that's interesting.
No, I know. I guess I should mention, I know Latiparus, so chicken of the woods. Most of what we have that grows up here grows on conifer and a lot of it on hemlock. And so I'd say there are a lot of folks who won't cook it well enough. You like, as far as my experience and all my mushroom friends, we agree with this, to bake it first, release a lot of the moisture in there, and then do something else with it, whether it's frying it, sauteing it, or something like that. It definitely has to be cooked really well. Otherwise, it usually does cause you maybe to poop your pants or something like that.
Okay.
Yeah, that's a common one I see around here.
Yeah, it's one I'm actually sensitive to.
Yeah, I hear a lot of people are, no matter what.
Even fully cooked.
I'm so sorry. I love
even
that.
even overcooked.
No.
Yeah, it's definitely for me not just the cooking. Although I would imagine that would make it worse. So I appreciate your mentioning that.
All right, we have one more from YouTube. Andrew would like to know if you keep any specific remedies around in case of consuming a toxic mushroom.
I don't. I am super cautious, and my remedy is that I have poison controls number on stickers that I got at a mushroom fair once, and I have those stickers on the inside of all my field guides. And that way I have it readily available just in case. But I also have a lot of friends that I can call. Honestly, if you don't know what it is, that's the best thing to do. Yep. But I've only ever had one experience where I've gotten sick from a mushroom because I am really careful.
Good advice.
Yeah.
Any other, excuse me, any other questions? Well, if not, I'd like
Related.
to. Go
Sorry.
ahead. Yeah, please.
I had one, not really related to mushrooms, but who did the illustration of the mushroom moon? That was really neat.
Oh, which illustration?
The mushroom moon, you know, the.
Oh, so a friend of mine, it was an idea that I had when I realized that the 50-year anniversary of the moon landing was happening at the same time as the 50-year anniversary of fungi becoming a kingdom. I thought that would make a really cool t-shirt. So I talked to a friend of mine who's a graphic artist. Her name is Sarah Graham. And she's in Canada, and you can probably find her on Instagram most readily. And yeah, I commissioned her to create that, and then we made t-shirts out of it.
Cool.
Yeah, they glow in the dark too, so it's cool.
You still sell the t-shirts? Or?
Yeah, I do. And this one too, this Mycelium one, my friend Travis made this. It's kind of like a metal mycelium.
Nice. Right.
Yeah, so I do. I have an
That's on your Etsy?
shop. Yeah, it's an Etsy, and it's all under Yellow Eleanor. And for mushroom people, you would appreciate some of the very cheesy mushroom pun related t-shirt ideas.
All right. Well, thank you. And thanks for the talk.
Yes, thank you guys.
Anyone else?
I just wanted to say thank you to Rachel for being so wonderful and sharing everything. And it's been really wonderful. We've been looking forward to seeing you here.
Thank you.
Yeah.
Well, thanks for your help. All right. Well, let's have another round of applause. Really appreciate your coming to LAMS and talking with us and getting everyone back in on the ground floor. So I think it really is helpful to orient people this way and to share your experience and passion.
Thank you.
Very inspiring. Thank you.
Thanks.
Any last words? And otherwise, I think we'll wrap it up for tonight.
Thank you.
Thanks, everybody.
Thanks again, everyone.
Thank
That's
you.
a great talk.
Great job.
Thank you.
Amazing. Thank you. You got the best shirt I've ever seen.
Okay, I'm gonna go now.
All right.
Thanks, guys.